It's really a great pleasure to be here. I've very much enjoyed the discussions over the past few days that certainly went beyond science. And so what I would like to do today is share with you some of the efforts that we've been putting in trying to recapitulate aspects of human brain development. And I'm a professor of psychiatry, and my main interest really has been in understanding the biology of psychiatric disorders. And if you were to think of the biggest breakthroughs that we've had really in psychiatric research in the past decade or so, is really the fact that we've been identifying hundreds of genes that are associated with psychiatric. And so, for instance, here you're seeing the top 100 genes associated with autism. And of course, the challenge at this point has been to understand what each of those genes are actually doing and how they're impacting the assembly of the nervous system, in particular of the human nervous system. And the reason why that is so challenging is that, to a large extent, human brain development is inaccessible. And by inaccessible, I really mean inaccessible at the molecular and cellular level in a way that would allow us to both dissect but also probe aspects of disease. And so, for instance, if you were to look at the stages of human brain development, well, we know quite a lot about the early stages of patterning and cell specification. And in the past decade or so, we've learned more and more about cell diversity across the nervous system. So now we have atlases of parts, at least, of the human nervous system. The reality is that the more we advance in human brain development, the least we know. And so, for instance, we still have a very poor understanding of how cells are moving across the nervous system, and this is more of a rule rather than an exception. And of course, we know very little about how cells are finding their final positions, how they're connecting with cells at very long distances, and perhaps one of the most puzzling aspects for human brain development is, the last schematic doesn't show up, is how some of this refinement and plasticity takes place in humans, especially because this has been prolonged for a couple of decades. I don't know if you hear that noise, it's me or, yeah. Not sure exactly what to do, but maybe I'll move it further, yeah? Is this okay? Towards me? Oh yeah, okay, well, that's paradoxical. So there are a number of ways in which you can access, at this point, some of the stages of human brain development. And what I've tried to actually do here is I've tried to classify some of what we could call brain surrogate models of nervous system development based on either biological complexity or genetic validity. And so really, there are at least four. On one hand, there are stem cell models that are based on iPS cells or embryonic stem cells that are differentiating in a dish. There are non-human primates, which certainly have a lot of the biological complexity, but less of, obviously, of the human genetics. Then there are ex vivo human tissue models where we can now keep fetal tissue or surgical tissue for longer and longer in a dish. And then somewhere in between are what we call chimeric models or transplantation models. So today I'll actually focus on the two on top, the stem cell models and the chimeric models to a large extent. And of course, we use some of the others, but I think it goes beyond like what we're talking today. And really, to a large extent, I guess the promise of stem cell models for studying psychiatric disease is started with the ability of reprogramming fibroblasts in this case, as Yamanaka has beautifully shown, into induced pluripotent stem cells, and then taking those induced pluripotent stem cells and differentiating them into neurons. And 15 years or so ago, we've derived some of the first neurons from patients with autism, which are really sitting at the bottom of a dish here. Certainly quite complex in terms of their activity, and you could patch them and do calcium imaging. The challenge was that we were unable to keep them for very long periods of time. And after about 100 or so days, they would essentially disintegrate and come off the plate. Of course, there's nothing in the context of human brain development which takes hundreds and hundreds of days. And so with the advent of three-dimensional cultures, it suddenly became possible to maintain some of these cultures for very long periods of time. And by long periods of time, I really mean hundreds and hundreds of days. And really, the other important aspect of this is that now, by keeping the cells in three-dimensional cultures, you can leverage not just self-organization, but mechanical forces, gradient, stochastic forces even, to develop cultures that are ever more complex. There's certainly been a lot of confusion about the many models that have been developed. So I had really the great pleasure of working with a large group of leaders in the field, Jürgen included here, in trying to reclassify and simplify some of these models, which certainly, in the context of the discussion that we've had before, it's very important in communicating with the public. And really at this point, we think that there are either two-dimensional cultures where you can make relatively pure population of neurons, astrocytes, and microglia, which we can put on a chip, or you derive three-dimensional cultures, which we really distinguish by the amount of guidance. So on one hand, you can use very little guidance and get cell diversity, and this has really been quite important in understanding self-organization and self-specification, or, and this is work that we've primarily done, has been to actually guide the differentiation to very specific brain regions, which we call regionalized neural organoids. And then if you want to study cell-cell interactions, then we can actually fuse some of these organoids 2 by 2 or 3 by 3 and make assembloids as preparations, or you can transplant them. And really, I think this is an important slide to some extent because I think it puts together some of the terms that I'm going to use moving forward. And so how do you make actually a regionalized organoid or a cortical organoid? This is a representation of the first protocol that we've developed where you can essentially take intact colonies of pluripotent stem cells, you can essentially dissociate them in single cells, and you can force them to aggregate in these tiny spherical structures, which we initially called spheroids. And then you pop them out of that plate and move them to a low attachment plate, where essentially they'll never be able to sit down. And when you use this very simple trick, what happens is that the spherical structures will continue to grow, reach about 4 millimeters in size, but actually continue to develop. And we've shown over the years that such a simple preparation actually recapitulates a lot of the cell diversity in the cerebral cortex, that they can go beyond just generating deep layers and upper layer neurons, but at one point they'll switch and they'll make astrocytes, pretty much at the time that you would expect them to make astrocytes. This is work that we've done early on with the late Ben Bears. That this process is highly reproducible across lines, and quite interestingly, of course, that you can run functional assays, and this has been very important for us, for me as a neuroscientist in running functional assays, which I'll get back to it. So one of the remarkable aspects or features of these cultures is that you can now keep them for very long periods of time. And we've maintained the longest reported cultures to date, going beyond 900 days in a dish, but in principle, you could keep them indefinitely. And in a series of papers, we discovered something quite remarkable. If you keep them for 900 days, in the first 9 months or so, they look very much fetal. And then I'm not sure whether you can see here, but somewhere around 280 days or so, they'll transition to a postnatal signature on their own. And this happens at the RNA level, it happens at the chromatin level, it happens at the methylation level. It even happens in terms of canonical switches that are very well known. So many of you probably know the NMDA receptor switch from 2B to 2A. This happens almost like a clock in this culture at about 280 days without us providing any cues about the time of birth. So you can even see it here, for instance, through electrophysiology and pharmacological dissection of this. So this tells us that once you start differentiation in a dish, there is an intrinsic clock of maturation that continues. And of course, that's both an opportunity, but also a curse when it comes to modeling much more complex late onset disorders of the nervous system. And over the last 10 years or so, we've actually been using these models to study, for instance, the role of astrocytes, together with Viviana, actually to identify viruses that infect specific cell types, to map disease genes, to ask questions about like human and chimp evolution by creating hybrid iPSLs, or to ask questions about disease, both environmentally derived, such as hypoxic encephalopathy or prematurity, or very common forms of genetic disease, such as 22q11 deletion syndrome, which is the most common cause of genetic cause of schizophrenia that we know of. So most of the work that I've shown you so far has been in looking at the cerebral cortex, which certainly is an important part of the brain, but not all the cell types reside there. And so how are we going to generate a lot of the other cell types in the nervous system? In fact, you may wonder how many cell types are there in the developing nervous system. And just in the last years or so, we've learned through a profiling of the first trimester human embryo, an entire nervous system, that there are at least 600 cell types and cell states in the human nervous system, if you take the entire nervous system. And so we wondered, could we actually generate a lot of the cell diversity? And I think we've discussed a lot about AI, but what you can actually do is you can actually ask ChatGPT, as you can see, that's me, SE stands for my initials, and you can say how to generate different cell types in neural organoids. And it will actually give you a beautiful answer where it tells you that to generate this, you start with iPSLs, you induce neural differentiation, you regionalize the organoid, and then you characterize the organ. Of course, the challenge is that if you read carefully, it says this may involve adding specific growth factors, small molecules, other signaling that are known to promote the development of a particular brain region. But that is the problem, that very often we don't know neither which ones they are, or if we do, we don't know when to put them in what combination. So two incredible physician scientists in the lab over the past few years have actually tried to take a very systematic approach and now take the main signaling molecules that pattern the nervous system and create a race screen where we can actually combine them 2 by 2, 3 by 3, and then run single-cell RNA-seq at the end to see how much cell diversity can we generate. And as you can see here in this UMAP space, you can very quickly, by just adding a few of these morphogens, move a cell type across this three-dimensional space. And if you now actually combine this with the previous atlas that I've shown you of the human nervous system, we've discovered that with just 48 conditions, we can actually generate 2/3 of the cell diversity of the nervous system at that stage. And we've very carefully annotated most of the cell types, so you can see there's thalamus, there's hindbrain, there are parts of the brainstem, and so on and so forth. In fact, we have even generated this very interesting cell type, which has just been described in the last 3 years, which is a primate-specific type of interneuron only present in primates and only in the striatum. It represents 1/3 of all the striatal interneurons. And one of the conditions that actually generated precisely that cell type, which we've been characterizing quite carefully. And this shows you that we're at a point where we can actually generate a lot of the cell diversity across the nervous system, either through directed approaches or, in this case, kind of like combinatorial approaches. So how about cell migration? How do we study cell migration in the nervous system? So 7 or 8 years ago, we came with a very simple approach where we essentially, to study, for instance, migration of interneurons, we generated either the dorsal or the ventral forebrain in organoids, and then we essentially put them together in close proximity to each other, just wondering what would actually happen. And it turns out that when you do this, the expectation was that interneurons, GABAergic cells, would actually identify the dorsal forebrain, point towards it, and start migrating towards that. And that's exactly actually what happened. And this is one of the first movies of that, where you can actually see all the GABAergic cells are labeled here in white. They're in the ventral part. And it's essentially within 2 weeks, they have invaded, almost like a tumor, most of the dorsal forebrain. And they do so by moving in a very specific way, by undergoing very stereotypical nucleokinetic movements. Why is this important? It's because we can actually start asking questions about disease. And this is, for instance, just for the beauty of the molecular biology, of the cell biology of it. It's one of these interneurons that it's undergoing this movement, which really in humans has not been observed before. It happens in the second, third trimester of life. But you can actually see how for about 30 minutes here, the cell will actually point out in the direction of the cortex and will essentially undergo nucleokinesis. So essentially the nucleus will be pulled up by actomyosin about 40 microns. And then we'll spend another 3 hours trying to decide the next step, and over again. So interneural migration has been hypothesized to be important for autism, mostly because of the excitation to inhibition ratio. But there are hundreds of genes associated with autism. How do we know which one of those are actually important? So a few years ago, we actually set up a screen, which actually just published like a month or so ago, where essentially we were able to screen more than 400 autism and neurodevelopmental genes to see how many of those genes are either impacting interneural migration or interneuron generation. And this required a lot of effort. So Shanling Meng actually had to make about 1,000 assembloids that she cut manually one by one, and then pull the cells by fact sorting, and essentially identified that about 10% of these genes are actually interfering with one step or another. And there's actually quite interesting biology that we've discovered. For instance, we found that one gene called LunaPark, which causes a severe epileptic encephalopathy, is actually causing a migration defect, but this actually is an endoplasmic reticulum gene. And Shanling, it's a longer story that it's published, so you can actually check it out, but actually she discovered, quite surprisingly, that the entire endoplasmic reticulum moves in front of the nucleus before the cell undergoes that jump. And then when patients carry this mutation, that actually fails to happen, and so the cells don't move. So we think there are fewer interneurons in these patients reaching the cortex, and therefore less inhibition. So how about connectivity, which I guess it's perhaps more relevant in the context of like a higher processes. So to do this, we've actually started building projection-based assembloids. So for instance, the first one that we built was of the cortical striatal cortical cells project into the striatum. The striatum does not directly project, but rather comes back through a loop. So to do this, Yuki Miura and the lab made a striatal organoid that has this beautiful medium spiny neurons in it, and then essentially fused it with the cortex. And so you can see the cortex here is in green, and the striatum here is in purple. And when you put them actually together, the cortex will project into the striatum, but the striatum will not project back. And in a form of neurodevelopmental disease known as Phelan-McDermid syndrome or SHANK3 deletion, we found that unless you actually have the cortex, you can actually not discover, you cannot reveal the phenotype in striatal cells because they actually need the glutamate input in this. So I think this illustrates that some of the cell-cell interactions are important for modeling some of these disease processes. Now, this is putting two brain regions, but of course, many circuits in the brain have more than two parts. So then we wonder, could we actually put three? And so a few years ago, we've actually tried to build the entire corticospinal circuit from parts, where the idea was that you would make a cortical organoid, you'll make a spinal cord organoid, and then you'll make a human muscle derived from a biopsy, and put them all together. And if this was to work, then deep layer cortical neurons in the cortex will find motor neurons in the spinal cord, and motor neurons will find muscle and trigger muscle contraction. And it turns out that this works really, really well, that without us providing any instructions, the cell actually find each other, and then you can actually put an opsin in the cortex, and then perform calcium imaging, where stimulating with light just the cortex. And this was early days when we didn't have microscopes. This is a large structure. This is 9 millimeters in size. So you're only seeing the muscle here. The spinal cord is on the far right, and you have to believe me that the cortex is even farther right than that. And essentially, you'll see that at one point it will go stim. This is a movie. And when it, which I hope will work, doesn't work. Oh, yes, it works. It will go stim, and that essentially triggers a muscle contraction. And in fact, quite interestingly, the more you do it, the more reliable this process is. And of course, you can do pharmacology to dissect, but this tells us that despite us not knowing the rules of assembly for the circuit, making the part is sometimes sufficient to build some of these first early circuits of the nervous system. Now again, most of the circuits in the nervous system don't have just three parts. They have 4 or more, and many of them are actually not linear, but rather in a loop. So for the past few years, Yuki has been very interested in building a loop circuit of the nervous system, which is primarily the direct pathway, which I'm sure most of you know, that involves cortical neuron projecting to the striatum, striatal neuron projecting to parts of the midbrain, the GPE, and then going back through the thalamus back. And so this is a longer story. Yuki spent several years actually optimizing the cell composition of each of these parts so that we make sure that we have the key cell types in this circuit. And this is a UMAP that has about almost 100,000 cells that shows us that we can generate the glutamatergic cells of the cortex, the GABAergic cells of the GPE, and some of the GABAergic cells of the striatum, but also the GABAergic cells of the thalamus, which are very different. So now with all this cell types, what do you do? How do you put all this together? And we thought it's going to be not that difficult to put 4 because we've put before 3. It turns out to be much more complicated than this because essentially every single effort of ours to actually fuse them has failed. Then at one point, we started actually building this 3-dimensional, simply 3D printed wells where you can position each of them. This was the first version, very ambitious. We thought we were going to put even 8. Certainly failed. They would always like essentially come apart. Then we had like version 3, which is slightly a little bit better. Anyway, 10 versions later, we finally had one where you could actually put all 4, keep them for about 48 hours, and then pop them out, and you will have these beautiful assembloids that you can actually keep now for hundreds of days after and study activity. And there's some remarkable properties of this because now we can actually do calcium imaging, and we've built, Yuki has actually built a macroscope that allows us to image this entire circuit. And so you can see the cortex, the basal ganglia, parts of the mesencephalon, and the diencephalon, or the thalamus, all 4 put together. And once you actually put them together, something quite spectacular happens. And essentially, the entire structure starts to synchronize its activity, which I'm not sure, you probably can see it better from this raster plot that actually shows that most of the activity is actually synchronized. Now, you can do even more than that, and we've built actually an electrophysiology rig that allows us to put electrodes from all 4 parts. And to be honest, this has been a dream of mine because I trained actually in Wolf Zinger's lab when I was a medical student. And I always dreamed of recording from human neurons one of the preparations. So what we've actually done, and I'm not sure whether the audio will actually work, but you can now actually listen to the activity of the loop as it actually travels through these 4 parts, which is certainly quite surprising to us because it's been very difficult to get relatively complicated structures. And now we have like about 9 or 10 genetic mutations that impact different parts, so you can actually build hybrids. and identify where each mutation is impacting what part of the circuit. So in the last few minutes that I have, I want to actually tell you about like even more advanced systems that we've been building to try to essentially map human circuits and reconstruct human circuits. And this actually was prompted by a very surprising observation that we made a few years ago. So this is a stem cell-derived human cortical neuron at about 200+ days kept in a dish. I mean, if you look carefully, it does look like a pyramidal neuron, right? I mean, pretty much has like a long apical dendrite. It has a few basal dendrites. It looks sort of pyramidal. Now, if you were to take human tissue, early postnatal, and you were to fill a neuron with exactly the same method, early postnatally, a cortical neuron would look something like this, about 9 to 10 times larger. So we started wondering, why are the cells, even when we keep them for hundreds and hundreds of days, they're not growing that large? Moreover, the resting member potential of most cortical neurons in vitro is about -40, -50, almost never -70. So very difficult to do most electrophysiological experiments. So then you could say that, you know, certainly in vitro reductionem, but in vivo veritas. And so then we thought, let's try to transplant some of this culture and see whether that would make a difference, not necessarily in the timing of development, but in the features of the cells. And so then we came up with this very simple approach, which we published last year, where essentially you would do exactly the same as you've seen before. You would grow this cortical organoid for about like 60 to 70 days, and then you take an intact organoid, and essentially you place it in an immunocompromised rat, but you place it intact right into the somatosensory cortex of the rat without removing rat tissue. This is an immunocompromised rat. It turns out that if you do this trick and you do it early in development, in the first week after birth, before the closure of the critical period, the organoid would actually grow quite large. Will grow so large, it would become about 1/3 of a rat hemisphere. And essentially will create, and that has been really our goal, to try to create a unit of human tissue in a very predictable position. And I hope you can see from this image of the rat brain, this cross-section, that now the human cells essentially represent the unit that goes all the way from the pia to the ventricle. Now, not only that this is not an inert tissue, nor is it like kind of like amorph, but actually if you look by single-cell gene expression, we've discovered that you have most of the cell types in the cortex. They will actually develop pretty much at the same pace. So if you use the Allen Institute Atlas, you can pretty much map, you know, very specific cell types in the cerebral cortex. You can also open the skull of the rat and now perform live imaging of some of the cells with the spontaneous activity of cortical neurons. Even some astrocytes, actually, if you watch carefully in the upper part, there's actually some astrocyte waves that are spreading throughout, which again are human astrocytes. 99% of the cells in the graft are human. The others are essentially endothelial cells that are essentially feeding the graft. So now the prediction was that if we put the graft in the somatosensory cortex of the rat, and if they were to receive thalamic input, you should be able to move the whiskers of the rat onto the opposite side and trigger responses from human cells. And this is exactly what Felicity has done. And she has actually found that within essentially a second of stimulation, which is what people have actually also discovered in the rat through calcium imaging, you can trigger these beautiful responses of human cells, but obviously not the same thing with randomized timestamp. And we think that this activity, this physiological activity that the graft is receiving, is responsible for this other interesting feature. Because now, if you take a cortical neuron that you kept in vitro in an organoid for 250 days, and an organoid from the same batch, same line of differentiation, but put into a rat, it would look like this after 250 days. It grows 6 to 8 times larger. Now it has a resting member potential of -70, -75. It has spines. It's much more mature and certainly integrated into a circuit. So although the timing of differentiation is somehow kept, there are some features of development that are missing unless you provide some of the in vivo environment. And now, the next question for us, and perhaps this was one of the most controversial experiments that we've done at that time, was that if indeed the cells are, the in vivo cells, human cells are integrated, would they be able to participate to behavior in the rat? So to ask this question, what we did is we delivered chanorodopsin, an opsin, into the cortical organoid before transplantation, washed out the virus, transplanted the human cortical organoids pretty much the same way we did for the rest, waited for about 140 days. The graft has grown. Now it's about 1/3 of a rat hemisphere. And then we implanted an optic fiber right on top of it, and then started doing behavioral training of the rat, more precisely reward conditioning. So we performed reward conditioning where we were teaching the rat to associate the liver of a reward with stimulation of human neurons by blue light. And it turns out that if you do this for about 14 days, the rats actually will learn to predict that the reward is coming after stimulation of human neurons. And actually, you can see this, the learning curve, but perhaps it's much more clear from this experiment. You can actually see the rat receiving red light, which is certainly the control, so nothing happened. You see the water spout right in front. And then at one point, you'll see that blue light will come. And essentially, in 99% of the trials, when we provide blue light, they will actually go and drink water. So they're not necessarily thirsty here, but this demonstrates that the human cells have been sufficiently well integrated into the rat cortex that they can actually even modify the behavior of the animal. And why do we want to do this? Well, because most psychiatric disorders are behaviorally defined, because many of the phenotypes for psychiatric disorders are subtle and at the circuit level. And in order to understand them better and actually to develop some therapies, in our opinion, we will need to get more complex models. And in fact, there are many, many applications of transplantation, and this actually overview is going to come up like very soon in the next few weeks. But essentially, you can transplant single cells, you can transplant intact organoids, you can transplant assembloids, which has not yet been published. But you can also envision that there are going to be approaches where you'll deplete parts of the rat or mouse cortex and transplant human cells. Or you could actually do neuroblastocyst complementation, where you would actually insert some of the human cells very early on and have human cells participate to parts of the nervous system. And certainly there are various degrees of ethical concerns for this and complexity, but most of the applications are, first of all, for obtaining advanced maturation of the cells, obtaining circuit and behavioral readouts, drug testing, and certainly cell therapy, as we have seen for Parkinson's disease, which seems to be quite promising. So let me show you how you've been using this to understand the disease, because I think this is the most advanced model that we've had so far, and certainly the most that has the potential for translation. And this is for a disease that I've now been studying for 15 years called Timothy syndrome, a very rare form of autism and epilepsy. We only know of about 50 patients worldwide. But this is highly penetrant, almost 100%. The patients have a single point mutation in a calcium channel, CAC9-1C alpha-calcium channel. And we've discovered 15 years ago that if you make cortical neurons from this patient, there's going to be more calcium inside the cells when you depolarize. If you make interneurons, they actually migrate in a very peculiar way. They jump much more often, but every single time they jump shorter distances, so essentially they're left behind. But there's another thing that we've discovered when we were looking very carefully. The mutation is actually in an alternatively spliced exon that is actually developmentally regulated. But not only that, the mutation sits right close to the acceptor site. So when the patients splice this exon, they actually make even more of the isoform that carries the mutation. So they amplify the phenotype. And we and others have tested the two isoforms, and they essentially are indistinguishable in terms of their functional capabilities. So then we thought, how about getting rid completely of this isoform as a therapeutic opportunity? And by the way, this isoform goes down with development, so in the adult, it's really the minor isoform. So then in the lab, we essentially run a step-by-step screen to identify an antisense oligo that would very efficiently take down just specifically that isoform. And it turns out to work really, really well. And Xiaoyu Chen, who's actually been working on this for several years, identifies several antisense oligos that you just add to the cultures, and essentially within hours, they'll take down the isoform, and within a couple of days, the protein will also be changed. And actually, the protein levels remain the same. And we've been able to rescue every single phenotype that we've discovered over the past 15 years. You can rescue the calcium phenotype by adding it to the cells. You can patch and rescue the inactivation of the channel. You can even make forebrain assembloids, fuse them for about 2 weeks at the antisense, and essentially rescue the interneural migration. So you may wonder, for a disease like this that is very severe, highly penetrant, for which there's no animal model that recapitulates the disease, what is the next step therapeutically? So you can actually ask again ChatGPT, and this is not me, but this is Xiaoyu who's asking, what would you do for a screen for Timothy syndrome? It actually will tell you, again, very, very well, what are the stages? And we'll say, identify the mutation for Timothy syndrome, done. Determine the target region for antisense, done. Design the antisense, done. Test the antisense in vitro, done. Then it says, test the antisense in vivo. And essentially, it turns out that it's really important to test this antisense in vivo, not just for toxicity, but because many of these antisense actually do not work in an in vivo environment. So how do we go about that? How do we test this in an in vivo environment if there is no primate model, no animal model of the disease? So we thought we could actually leverage the transplantation platform. So what we did is we transplanted now patient cells from 3 patients into the rat, grew to have 1/3 of a hemisphere to be human patient-derived, and now injected the antisense intrathecally, as you would do in patients, and looked about a week later. And it turns out that immediately the isoform goes down, protein stays the same, the calcium imaging phenotype is immediately rescued, that delayed inactivation, and there's also, and I haven't shown you, there is a dendrite retraction that is activity-dependent in Timothy syndrome. And actually, within 10 days, you can get complete rescue of the dendrite phenotype in this patient in vivo. So at this point, we've actually went on and identified most of the patients. And with one of the child neurologists who's working in my lab, we've actually talked to most of these patients. And we've been working to get ready for a potential clinical trial as soon as we finish the toxicology studies that we're doing. So anyway, this is just to wrap up and hopefully convince you that it's really an exciting time for human neuroscience as we're getting closer and closer to having models that recapitulate not just aspects of human brain development, certainly in isolation, not in total, but also predictable model that hopefully will derive therapies. And this has really been a group effort, and I must say, I think there's been some discussion about this. We've put a lot of effort into sharing the technology. So we have, like, my center at Stanford has a workshop that has been training now close to 200 plus labs around the world to implement some of these technologies. And certainly this has been one of the most rewarding part of, and this is like the taking of a selfie that we've been doing. And with this, I would like to thank you and glad to take